Okay, so this is our S1P2 lecture, which will deal with some chemistry information, bonds, functional groups, and biomolecules. Again, this is information for lecture exam 1. All right, so first thing we'll talk about is chemical bonds. First of all, before we talk about bonds, let's undergo a few definitions. A molecule is two or more atoms joined together chemically. This joining together is referred to as a bond. One of the problems with bonds is that oftentimes we learn about them in earlier classes where you get this idea that it's some sort of physical structure. As an example, in high school, somebody brings in toothpicks and gumdrops, and you take, a yellow gumdrop and put two white gumdrops on it. So I just made a water, right? And that water molecule, right, is held together by these toothpicks. Those are the bonds. Bonds aren't physical things. They are just really a favorable energy relationship. So it's more favorable for the bonds to stay together and hold the molecule together than it is for them to come apart. In this course, we're concerned about 4 basic types of bonds. The covalent bond, which has 2 subcomponents, polar and nonpolar, an ionic bond, and then a hydrogen bond. The chemical formula of the molecule held together by bonds indicates the number of each atom that makes up the molecule, and from that we can calculate the molecular weight. So as an example, right, water has a chemical formula H2O, right? So it's got 2 hydrogens and 1 oxygen. If we wanted to calculate the molecular weight, we'd have to know that hydrogens count for 1, right? So they have 1 Dalton or 1 AMU. Since there's 2 of those, that's a 2. And then oxygen is a 16. And there's 1 of those, so 16 plus 2 is 18. So the molecular weight of water with the 2 hydrogen and the 1 oxygen would be 18. So let's look at the different bonds. Let's talk about covalent bonds. When you see the word covalent, you should think about sharing electrons. And covalent bonds are about an equal sharing of electrons. When atoms pair together, then they decide to share electrons. And how much an atom shares an electron really is determined by its electronegativity. So as an example, hydrogen has 1 electron typically. And if you have 2 hydrogens that come together and they each share each one other's electrons, right? So if they want to share, they can share each other's electrons. So here's a hydrogen, and this little dot represents an electron. Here's a hydrogen, and this little dot represents the electron. So they share each other. So this hydrogen has 2. This hydrogen has 2. And that forms a H2 molecule, and that's typically how we see like hydrogen gas. Now, in the case of hydrogen, since it's the same molecule, they have the exact same what's called electronegativity. And it's the electronegativities that really determine how much of an electron does a specific atom hold on to. And so atoms with high electronegativities hold on to their electrons a little greater, and they tend to be a little more negative. And so it really depends on the electronegativities of the different atoms that bind together. When we have the 2 atoms that are exactly the same, in this case both hydrogen, they're going to have the same electronegativity, so that won't matter. Okay. So remember, for hydrogens, the outermost shell, right, would be 2 electrons, so they would become more like helium, if you remember in the upper right of the periodic table. Then each hydrogen has its electron shell filled, and it's happy, and it tends to be a little more stable. So you can look at the different diagrams, and I find that the Lewis dot tends to be fairly easy to, the one right here in the middle, to decipher. But just as long as you understand it, I'm not going to worry about, which one of these methods you learn. So whatever you find is the easiest for you to remember these things, and that's fine. Again, since hydrogen atoms are the same atoms, they're sharing electrons equally. And if you share the electron equally, it means you're just as likely to find an electron at one point of the molecule than you are another. And so, since there's no charge distribution, that's referred to as a nonpolar. So see it down here, nonpolar covalent bond. Now, there's other examples of nonpolar covalent bonds, and one of those is methane. Okay? So methane, even though it's made up of different atoms, it's one carbon atom with 4 hydrogens bonded to it. Okay. Again, if you look at the periodic table, carbon would have 4 electrons in its outermost shell. Hydrogen, we established before, had 1. And so each carbon shares one of its electrons with a hydrogen, and each hydrogen shares one of its electrons with carbon. So carbon has 8 on the outermost, which makes it more stable. Each hydrogen has 2. Despite the fact that these are different atoms, carbon and hydrogen, they have relatively similar electronegativities. It means that the electron is not more likely to be found at any point of this molecule than any other place. So the electrons are shared equally. So this is an example of another non-polar covalent bond. So carbon and hydrogen share electrons very equally, and therefore they tend to form non-polar covalent bonds. This is important, so don't forget that detail. Okay, and carbon and hydrogen share very equally, and that becomes really important when we start looking at biomolecules and how they're arranged and other things like that. Now, water is a little different. We mentioned water, right? It's got that chemical formula H2O. And when we look at an oxygen molecule, right, an oxygen has 6 carbons in its outermost shell. So there's the 2 there. Don't worry about the inside 2. Here's 2. And then it's going to share one of its extra ones with hydrogen. It's going to share its other extra one with hydrogen. Hydrogen, as before, is going to share each one. So when we look at this oxygen, it's going to have 8 in its outermost shell, and that's going to make it stable. Look at this one, hydrogen. It's going to have 2. This hydrogen is going to have 2. And it's a fairly stable molecule. The problem is oxygen is very electronegative compared to hydrogen, and it has a strong attraction to the electrons. So when hydrogen shares its electrons with oxygen, we're more likely to find an electron around oxygen than we are hydrogen. Okay? Since they're sharing, it's called a covalent bond, but since they don't share equally, we're more likely to see, again, an electron around oxygen, so that becomes slightly negative, while the hydrogens become slightly positive. Since there's a charge distribution, this is referred to as a polar covalent bond. And polar tells us that the electrons are not equally spaced around the molecule. Okay? So this is a polar covalent bond. Now, the way you represent that with kind of shorthand notation is these little Greek symbols, deltas, right? So these little kind of weird looking S things, right? It means small. And so you can just write negative or positive, that's fine. And we write negative here because remember this is the water molecule, right? So this is our central oxygen and the 2 hydrogens. So the red larger ball is the oxygen. And it's got a little negative because remember we said that it's more electronegative and it's going to hold on to more of its share of electrons. And since electrons have a negative charge, that means overall the oxygen molecule is going to have a slightly negative charge. The whole molecule is neutral, right? But if the oxygen is slightly negative, that's going to make each one of these hydrogen molecules, right, slightly positive. So, they're not nearly as big a charge as the positives or negatives you'd find on an ion, right? So a cation, which is positive, and an anion, which is negative, have very large charges. But here, these charges are much smaller. But it does make the molecule polar. And because it makes it polar, it behaves differently than a nonpolar molecule in many cases, right? Now, atoms can share more than one pair of electrons, and we also have what are called double and triple bonds. As an example, O2, when it's formed, forms a double covalent bond, right? Just as a quick aside, do you think this is going to be a polar or nonpolar covalent bond? Give you a second to think about it. Answer it in your head. So I think one way to think about it is to go, okay, is one of these atoms more electronegative than the other? Well, no, since they're both oxygen, even though they're both electronegative, right, they are the same high electronegativity because they're going to bind to each other, which means there's going to be no charge distribution. So this is going to be a nonpolar covalent bond. Now, ionic bonds are an unequal sharing of electrons. And to form an ionic bond, the first thing you have to do is you have to have ions. And without getting into too much detail, and again, chemistry is prerequisite, if we take a look at sodium, right, sodium has one electron in its outermost shell. Okay? And atoms tend to be much more stable when they have a full complement of electrons in their outermost shell. And for our purposes, it's usually going to be 2 if it's hydrogen, and 8 for most of the other atoms we're going to worry about, those elements that are very different on the periodic table, don't have a whole lot of functional significance. So in physiology, a lot of those ones we don't have to worry about. As a matter of fact, there's really, the major atoms that we see in terms of the human body would be, what they call CHOMPs, CHONPS. So carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur are the big ones. And then also we see things like sodium, potassium, chlorine, calcium, magnesium are all fairly important. And there's some what they call trace elements like selenium that are minor importance, especially, they're important physiologically because you have to have some, as an example, selenium. And if you don't, your brain doesn't function correctly, and people with not enough selenium actually basically go crazy. What's interesting is if you have too much selenium, it also affects your brain, and those people go crazy. So having just the right amount of those, what they call trace elements, is very important. But we're really not going to talk about selenium. So there's not a whole lot of elements we're going to have to worry about for this class. And, certain elements, because of their position on the periodic tables, tend to form ionic bonds, and those tend to be at the extreme ends, except for that last column, right? The last column is the noble gases, right? And so that last column, it's column 8, if you're looking at the periodic table, has very stable elements in it. And that's because they have a full complement of electrons in their outermost shell. So sodium has one extra electron, and that means sodium's pretty reactive with that extra electron. It would like to get rid of it. Teleological statement going back from the first lecture, right? It would like to get rid of it so it would become more stable. Chlorine, right, has almost a full complement of electrons. It's just missing 1. Right? So it's got 7 in its outermost shell. They both would like to have 8. So if sodium gave its electron to chlorine, it would have 8 in its outermost shell now, because this shell would no longer have any electrons in it. And the 7 plus 1, right, would have 8 electrons now, and that would be more stable. Okay? So we can form an ion by giving up an electron. Sodium becomes positive because it gave an electron, a negative charge, to chloride. Chlorine originally. Chlorine becomes chloride. That's what we call chlorine when it's an anion, because it has an extra electron. So sodium and chloride now, being a cation, a positive charged ion is called a cation, a negatively charged ion is called an anion. They have what's called electrostatic attraction, and this electrostatic attraction forms an ionic bond. Okay, so it's an ionic bond is the electrostatic attraction formed between two or more ions. All right, so obviously they have to be oppositely charged, and typically they have the total net charge is usually neutral when we form these bonds. So, a great example of that is sodium chloride. Sodium chloride, I think most of you know, is a salt. Other types of atoms that form through ionic bonds would be bases and acids. So all acids, bases, and salts form by ionic bonds. So they're going to be formed by ions. Also, we mentioned this in another section when we talked in lab. That's how I kind of connect lab to lecture sometimes. Ionic bonds tend to dissociate, break apart when they get into water. Okay. Now, coming back over here to this whole idea of forming ions, students don't usually have a problem understanding what happens with chloride, right? Because chloride gets a negatively charged electron. Now the whole molecule becomes negatively charged. But sometimes students have a problem with the sodium. Sodium gave up an electron, so now sodium is positively charged. And I used to try to explain it by saying, sodium got rid of something that it didn't want, so now it's positive. Like, you'd think, hey, somebody paid your tuition and that would be positive, right? And that really didn't connect and work out for students. So I tell a stupid joke, and it's stupid, I admit it, but hopefully I'll remember, and maybe, one day you're sitting there on an exam and go, I remember that stupid joke, it's this. And so stupid joke is these two atoms walk out of the bar, and the one atom starts checking his pockets, and his atom friend says, did you leave your wallet? And he goes, no, I think I lost an electron. And his atom friend says, are you sure? And he goes, yeah, I'm positive. So yeah, you remember you lose an electron, you're positive. Stupid joke, yeah. And I won't even tell you that one that's even stupider about neutrons that don't pay for drinks because there's no charge. So, but that's forming ionic bonds when two oppositely charged ions or more are attracted to each other through electrostatic attraction. So there it is. Now, what's kind of interesting about, especially, ions and stuff, is this is elemental sodium here in the bottom left, and it's pretty reactive. You actually keep it in oil because if it touches water, it'll explode. This is chlorine. That's a nasty gas, right? We use that to disinfect pools and stuff like that. And by itself, if you ingest either one of these, you'll die. But as ions, when they form this lattice network and an ionic bond to form table salt, it makes your food taste better. So it's kind of neat. Chemistry is kind of neat that way in terms of that. So again, an ion is an atom that's now charged, that lost or gained an electron. If it gained an electron, right, it's called an anion because it has a negative charge. If it lost an electron, it's called a cation, and it has a positive charge. Right? The last type of bonding we're going to talk about are called hydrogen bonds. Hydrogen bonds require a hydrogen covalently bonded to an electronegative atom. For our purposes, from a physiological standpoint, that's going to be oxygen and nitrogen almost always in our course. Yeah, if you remember from chemistry as an example, fluorine, right, is very electronegative. It's also very kind of strange, behaves strangely. We're not going to worry about fluorine. Yeah, if you take it and you make it an ion, it becomes fluoride, and it can make your teeth stronger. But from a physiological point of view, we're not going to talk about it. So for our purposes, the two large electronegative atoms, physiologically important, are oxygen and nitrogen. And because of the reason that they hold on to their electrons a little more, because of that high electronegativity, we get that charge distribution within the ion, within the atom, where the electronegative portion, the oxygen and nitrogen, has more of the electron around it, so it's slightly negative, while the hydrogen has less and it's slightly positive. So a hydrogen bond can form within a molecule or between molecules. And here's a case of between molecules. This also happens in water too, but here's an example with ammonia. So here's our water molecule, right? Remember, the oxygen is slightly negative and the hydrogens are slightly positive. Well, the same case occurs in ammonia. Ammonia is NH3, so it's just nitrogen with 3 hydrogens around it. And because nitrogen, like oxygen, is a fairly high electronegative atom, it holds onto more of its electron. So it's slightly negative here. And the slightly negative hydrogen, the slightly positive hydrogen, positive here, right, has a weak electrostatic attraction to the slightly negative nitrogen, and it goes ahead and forms this hydrogen bond. Hydrogen bonds are very weak. They're easy to break. And we'll go back to this slide, just talk about it for a second more. And they're easy to break, but that you can form a bunch of them, all right? And so let's kind of look at a water bottle, all right? Now, we're going to assume that your water bottle that you're drinking is just pure water. And let's make it really easy and say it's a liter. Okay. Now, if you have a liter of water, right, how many mLs of that? I want you to kind of think for a second. Right. So 1,000 mLs is in a liter, so you have 1,000 mLs. All right. So I want to know how many moles of water is in that liter water bottle. Okay. And the reason I want to know is I want to know how many water molecules are there. Okay, so how do I know that? Well, let's see. Earlier in this video, we figured out what the molecular weight of water was, right? We said one oxygen is 16, each hydrogen is 1, so that's 18. So by definition, right, 1 mole of a substance will be its molecular weight. So if we have 1,000 mLs, right, divided by 18, that gives me almost 56 moles, right, 56 moles of water in there, right? Remember, a mole is Avogadro's number, 6.02 times 10 to the 23rd. So 55 times 6.02 times 10 to the 23rd, right, will be a very, very nice number, right? And it will be 3.3 basically times 10 to the 26. So you've got that many water molecules in a liter of water. Okay, now most people are half a liter, so that's only 1.7 times 10 to the 26 molecules. Okay, that's a lot of water molecules, and that's all sitting in this little bottle floating around. And so those water molecules, every one of them has just this charge distribution. So the one water molecule here, right, where this oxygen has this slightly negative charge around it, has a water molecule here, here, all over the place, right? And each one of those is forming a hydrogen bond. And so if you have enough hydrogen bonds, then you can form a fairly significant surface that has what's called surface tension. And a great example of surface tension would be if you've ever, jumped or dove into a pool or body of water the wrong way, right? You land on your back or on your stomach and it hurts a lot. And that's because of the hydrogen bonding is weak individually, but strong collectively, and it hurts. And so hydrogen bonding, does play a role. When we look at DNA, right? So I said, okay, what shape is DNA in? Almost all of you would invariably say, well, it's in a double helix. Well, what holds that double helical shape, that molecular structure, is hydrogen bonding within the molecule. So it's very important in terms of, forming molecular shapes, and it's, important in terms of how water behaves. And so, this hydrogen bond, while weak bond, has some important, physiological implications in terms of it. So that's the hydrogen bonds. So which type of the bond is the strongest? Well, we just finished talking about hydrogen being the weakest. We also said ionic bonds break up in water, right? So covalent bonds don't, right? So the stronger bonds are the covalent bonds, and then within the middle would be the ionic bonds. So here's a nice summary of, the three type of bonds, and they're only, doing covalent. They're not looking at polar and non-polar because there's no difference in the strength of those. While in the ionic bonds is in the middle strength, and the weakest are the hydrogen bonds. And hydrogen bonds really then are a consequence of polar covalent bonds. Because if a molecule wasn't a polar covalent bond, then it wouldn't form hydrogen bonds because it wouldn't have that charge distribution. Okay, so those are the bonds. Another important area of this is the functional groups. And functional groups allow us to understand how atoms behave, and allows us to understand why certain molecules are polar or non-polar, why certain molecules can move across the cell membrane where other molecules cannot directly. So we'll go over some of the more important, physiologically important ones. So the ones important. So the first one is called the hydroxyl group. The hydroxyl group is represented by an R with a bonded OH. Okay, so sometimes just go OH. The R in the old days when I was in school stood for the radical. And the radical was basically just the rest of the molecule. And nowadays, with us trying to simplify things, there's really no reason to call it the radical. So now most chemists call that R the rest of the molecule, right? So that's what it pretty much stands for. And so when you have a hydroxyl group, you're going to have this OH on it. And, this OH makes molecules that have it polar. All right, and these molecules tend to be polar because of the distribution of the electrons, because oxygen, right, has that strong electronegativity, so it's going to have more of the electron, so it's going to be polar. And it's also going to allow us to form hydrogen bonds, right? So the hydroxyl group allows us to form hydrogen bonds. So as an example, ethanol, right, and all alcohols, right, tend to have the OH at the end. Okay? We also see these with sugar, and that OH molecule allows sugars, carbohydrates, to dissolve in water. So that becomes important functionally. So carbohydrates can, like glucose, right, which is the most important carbohydrate in the human body, right, can dissolve in blood and be carried to cells for energy. So I already mentioned this, but we'll kind of look at it. What kind of bond forms between oxygen and hydrogen? All right, in this case, it'll be a polar covalent bond. Right? Polar covalent bond. So the next functional group that's important are the carbonyl and the carboxyl. The carbonyl and the carboxyl really aren't very different. The difference is the carbonyl occurs within a molecule, typically, not always. If it occurs within, it's a ketone, and if it's at the end, it's an aldehyde. But we're going to worry more about the carboxyl group. And the carboxyl group has on a carbon a double bonded oxygen with a hydroxyl group on it. Okay, and so these carboxyl groups are acidic. And the reason why is an acid is any substance that donates a hydrogen ion to a solution. Okay, and so the reason why these are acids is this OH at the end, okay, gives up its hydrogen. So let's look down here at the bottom right. So, okay, here's the chemical formula for acetic acid. It's, when you see, the word acetate or acetic, typically refers to two carbon chains. So here's two carbons, all right? And the purple outlines the carboxyl group. The one carbon has a double bonded oxygen with another oxygen bound to that carbon and then a hydrogen for that. When that hydrogen comes off, right, it becomes H+, and this oxygen molecule, right, becomes negative to balance the charge. Now that we have a free H+ hydrogen ion in the solution, that makes the solution acidic. Okay, so when it has the hydrogen bound, it's called an ic acid, like acetic acid, or we'll talk about pyruvic acid or lactic acid. All of those is when the hydrogen is bound. When that hydrogen diffuses away, it comes off, it dissociates and diffuses away, then it's often called an ate. Okay, so acetic acid becomes acetate. Pyruvic acid would become pyruvate. Lactic acid would become lactate. To be honest, for the purposes of our class, and I mentioned this before, we sometimes take shortcuts in chemistry where chemists would sort of say you're not supposed to do that. And we're going to assume that the acetic acid and acetate are pretty much the same. So I think, again, to kind of, ask a question, what is the end product of anaerobic glycolysis in humans? Okay, that's a question that many of you know, or when I tell you the answer, oh yeah, I did know that. That would be technically lactic acid. But sometimes people go, well, it's lactate. And technically it's not, because lactic acid is the end product. And then when that hydrogen dissociates, then it becomes lactate. When does that hydrogen dissociate? Well, at physiological pH, 99.7% of the hydrogen ion immediately dissociates from lactic acid. So 997 out of every 1,000 molecules of lactic acid we make immediately become lactate. So I think it's okay calling it lactate, because that's what's hanging around in your muscle anyway, or whatever cell was making it. Okay, so the carboxyl group is the acidic group. It's kind of a way to think about it. All right, and again, this hydrogen can come off as an H+. By definition, then, since we're donating an H+, any carboxyl group is an acid. Not surprisingly, then, carboxyl groups are found in fatty acids, amino acids, we'll see what the other side of amino acid is, and many other organic acids. So that's important. So we just mentioned amino, right? Here's the amino group. The amino group is the nitrogen, right? Because of where the nitrogen sits on the periodic table, it has one bond to the R group, the rest of the molecule, and then two hydrogen. Okay, so the amino group is an NH2. It's a nitrogen atom bonded to two hydrogen atoms on the carbon skeleton. Okay, so this makes amines. Okay, so things that are in general called amine. And not surprisingly, it's half of an amino acid. Since the previous slide, we said, oh, carboxyl groups are acids, not surprisingly then, an amino acid has an amino group on one side, that's the purple, nitrogen here, with the 2 hydrogen bound to a central carbon, and then the acid part, right? That's the carboxyl group, a carbon with a double bound oxygen to the OH on the other, right? This is the simplest amino acid that we have, right? And it's actually just to this, what they call central carbon here, it's got 1 hydrogen bound, that's called glycine. There are much, much more complex amino acids that have much more what they call side chains, areas here that are not just hydrogen. Just like the carboxyl group, it can become, oh, sorry, I don't want to change yet. It can be, the amino group can be ionized, but in this case, if it's ionized, it picks up an extra hydrogen and becomes a positive, right? So the nitrogen in this case acts as a base because it removes hydrogen from the solution. And any compound or molecule that removes H pluses from a solution is considered to be a base. So when it's ionized, it will have a charge of +1. Another common functional group we see is called a phosphate group. And a phosphate group typically has a phosphate bound to some central carbon, although it's not a phosphate directly, as we'll see in a second, with 4 oxygens. All right. So we'll see the phosphate here, and it's usually got an oxygen that's bound to the rest of the molecule, and then it's got a double bond in oxygen, and then 2 oxygens that are anions as well. So phosphate groups typically have a charge of -2. All right, so when we see these, they'll have -2. We see these in organic phosphates and things like that. And really the big places we see it are in the nucleotides. Okay, ATP is a nucleotide, right, as is ADP, right? ATP stands for adenosine triphosphate. So the tri is 3, so ATP would have 3 phosphates on it, not just 1. And DNA and RNA are made up of nucleotides, so they also have phosphate groups on them as well. Okay? Because this is negatively charged, it's going to be polar. As a matter of fact, let's kind of go backwards. The amino group, right, we said it becomes a positive charge, right? And so that's going to have a +1 charge. That's going to be polar. The carboxyl group is going to make an acid, right? We said that's going to be a -1 charge. That's going to be polar, all right, as well the carbonyl. The hydroxyl group can lose the oxygen, right? And that makes these polar as well because of the electronegative oxygen here. So that's polar. So every one of these functional groups we've talked about so far are polar molecules. Okay? Now we get to the methyl group, and the methyl group is very different. Okay? Methyl group is a carbon attached to the rest of the molecule with 3 hydrogens surrounding it. So here's a great example of a methyl group. It could also be in the middle of a molecule with just 2 hydrogens surrounding it attached to something else. Okay? So methyl groups are interesting for a bunch of reasons, but remember earlier we said that when carbon and hydrogen combine, that both have fairly similar electronegativities, which means that we are just as likely to find that electron around carbon as we are the hydrogens. That wasn't true with any of the other functional groups we talked about. Okay, so that makes this functional group held together by non-polar covalent bonds. There's no charge distribution, and so if it's non-polar covalent bonds, right, this methyl group makes it behave very differently. And basically, we see these methyl groups in steroid hormones. We see these in fatty acids, Okay, we see, so fatty acid, is it polar or non-polar? And you say, well, it's an acid, it should be polar, right? But a typical fatty acid in the human body might be somewhere in, 16 to 32 carbons long. And so it's got, one little acid ending, but it's got a whole bunch of methyl groups. So by the way it behaves as a, polar molecule. So methyl groups are very important and very different than the other groups, and it's a non-polar functional group, and we see this in lipids. Okay. So what kind of bond forms between carbon and hydrogen? We'll take a look at these. Since there's no charge distribution, right, it's a non-polar covalent bond, right? So non-polar, there's no charge distribution. We're just as likely to find an electron around any portion of the atom. Okay. So would a molecule with lots of methyl groups, right, mix well with water? So one of the things you probably remember from chemistry, and certainly, if you don't, I can remind you, that there's a saying, which is mostly true, like tends to dissolve in like. So polar things tend to dissolve in polar things, and non-polar things tend to dissolve in non-polar things. Since methyl group, right, is a non-polar molecule, right? The more methyl groups you have, the more non-polarness you have, right? And we said earlier, water, because it's a oxygen bound to 2 hydrogen, it's got a charge distribution because it's held together by polar covalent bonds, right? That the methyl groups don't mix very well with water and often precipitate out, so or at least aren't solubilized. So when you look at mixing water and oil, right? Take olive oil, pour it in water, right? The water and the oil don't mix, right? As a matter of fact, the oil just kind of sits on top, and that's because the oil is made up of primarily methyl groups, while the water is a non-polar. Sorry, water is a polar. So the methyl groups make the oil non-polar, while the polar covalent bonds holding water together make the water polar. So water is a polar molecule. All right, so let's stop and start a new file for the organic molecules.